Thank you very much. I want to thank the organizers for the invitation to come speak to you today. It's just my pleasure. It's an honor for me really to participate in this. My talk will be somewhat different than the earlier ones. Basically, the first thing I'm going to do is I find usually in an audience like this, a lot of people aren't even that familiar with plant diseases in general. So I'll give a brief overview of that, and then I'll talk to you about antibiotic use in plant agriculture. So plant diseases are caused by many different types of organisms, fungi being the most common, but there are plenty of bacterial diseases, viral diseases as well. Bacterial diseases occur on most crop plants that are grown. Most fruits and vegetables, there's some various bacterial problems that they have. They're very difficult diseases to control. And the main issues from these diseases is that they cause problems or spots or things that occur on fruit or vegetables, for example, and that would make those commodities unsellable in the fresh market. So that's one of the huge issues. Secondary issue with bacterial diseases is that they can kill plants, and that's always a problem, especially if we're talking about trees and trees are being killed. So here's just some examples of some diseases you may have heard of, citrus canker being one that has been a significant issue in Florida in the citrus production. It's caused by a Xanthomonas bacterium. You can see the lesions on the orange there. Bacterial spot of tomato and pepper is another serious issue. This causes probably 10 to 20% yield losses in tomato production on a yearly basis. This is another problem, bacterial brown spot of bean. This illustrates another aspect of bacterial diseases that they can cause lesions on leaves, and that leads to reductions in yield. So bean is not a crop that we use antibiotics on, but this is just a photo to illustrate the issues that we deal with. There's a lot of bacterial diseases that cause lesions on leaves. That reduces photosynthesis, obviously, and that leads to yield reductions. And then finally, here's some more wilt and rot type symptoms. Nobody's going to eat that potato, for example, I wouldn't think. But there's a lot of bacterial, not a lot, but several pathogens that enzymatically break down tissue like that, causing that rotting symptom. But for the most part, bacterial diseases are population driven, so they're very difficult to control. Because the population sizes can be quite large. We can see 1 million bacterial cells per gram of leaf tissue, as high as 10 to the 11th, which is an astronomical number of bacteria in the xylem fluid. So that's the water-conducting tissues of the plant. So these bacteria can build up quite large populations. These large populations then are what drives diseases. And I think the major issue that we have in trying to control plant diseases is that we have to try to control them in a preventative basis. We need to reduce populations or not enable the population size to build up to these levels. Once the populations are this high on plants, then the disease can't be controlled. It's just impossible to control. Now, probably for 100 years, we've had copper bactericides for bacterial disease control, and coppers have been used on numerous crops. They are fairly effective, but one of the issues with them is the coppers. They can provide a reduction in bacterial population size, maybe 10 to 100-fold reduction, but then those populations rapidly rebound. So coppers, over the long term, have not been super successful in bacterial disease control. And so it was recognized a long time ago, 50 or more years ago, that other agents were needed for controlling diseases. And another thing I want to say before I get into this further is when we talk about plant agriculture, one of the biggest issues that we face is the lack of host resistance in the plant. So many plants are resistant to diseases. They possess genes that code for proteins that recognize a specific pathogen that causes a disease, and then the plant will turn on a resistance response, and then that will prevent infection from occurring. Unfortunately, when we think about fruits and vegetables that we like to eat. Most of these are grown, the cultivars that produce these particular types of fruits that we like are usually highly susceptible to diseases. And so, for example, in this photo I'm showing here, there's 2 rows of apple trees. One row is Gala, one row is Golden Delicious. They're treated exactly the same way. In fact, there was no treatment in this orchard. And it's a little difficult to tell with this lighting, but the Gala trees on the left side have fire blight disease. So almost all the shoots on this tree were attacked by the fire blight pathogen, or amylovora. Whereas on the right side, the Golden Delicious trees, there's no disease at all. So by growing, for apple growers, for example, by having to grow cultivars such as Gala, Fuji, cultivars like that are very popular with consumers, they're growing varieties that are most susceptible to disease. So they're starting at a complete disadvantage already in trying to control diseases. So that's one potential mechanism or method that we have for disease control that we don't really have, because growers are forced to grow and produce varieties that consumers want, and the consumers want varieties like this that are most susceptible to disease. So in terms of antibiotic use in plant agriculture, the most commonly used antibiotic is streptomycin. And the streptomycin, of course, discovered in 1943, active against many different pathogens. At the time that streptomycin was being developed, it was also being tested for activity against plants. Again, so back in the 40s, as antibiotics were being discovered and developed, and they were being tested for control of human diseases, tested for control of animal diseases, they were also tested for control of plant diseases. In fact, penicillin, streptomycin, aureomycin, chloramphenicol, oxytetracycline, they were all tested on plants as well for control of various diseases. Luckily, really, I think streptomycin proved to be the main antibiotic that was effective, and these other ones, only oxytetracycline, has also had some use in plant agriculture. Now, some of the problems that were noted at the time and that kind of limited use of antibiotics, besides efficacy issues, was a lack of efficacy at lower doses, and then some phytotoxicity problems. And this is an apple tree here showing a streptomycin injury on the leaf with the yellow ring around the outside of the leaf. That's streptomycin phytotoxin. There were worse phytotoxicity symptoms seen, some leading to plant defoliation, which is a huge issue, has a strong effect on yield. And then the expense of these antibiotics at the time was thought to be too high compared to other existing methods of disease control. And so that kind of limited also the incorporation of antibiotics into plant agriculture. Now, streptomycin has been used commercially since the 1950s. What I want to stress in plant agriculture is it's only used on a limited number of diseases, only a limited number of plant hosts. So the main host for streptomycin use is apple and pear, and against a disease called fire blight, which I'll talk about later. Streptomycin is also used on celery. It can be used in nursery tree production to prevent a bacterial disease that kills shoots of trees, and then against bacterial spotted tomato and pepper. So the reality is there's a limited number of diseases that strep is targeted for, and the main one being a disease of apple and pear. It's usually used in a 100 parts per million solution. Streptomycin use on apples, it's difficult to get statistics for antibiotic, fungicide use in plant agriculture, and these stats are only up to 2003, coming from the USDA, National Agriculture Statistical Service. You can see that the usage is in the thousands of pounds, ranging from about, say, 10, 12,000 pounds a year to just under 25,000 pounds. And I would say after 2003 that range is probably similar. And usage of streptomycin, So the average would be about 20,000 pounds per year, and the treated acreage being about 15% of all apple acreage in the U.S. And so there are several regions in the country where fire blight disease is not as important of a problem as it is here in the Midwest and in the East. So I'd say the usage in the Midwest and Michigan, for example, and in the East probably a bit higher, maybe on the order of about 20 to 30% of the acreage. And generally, growers will put on 1 to 3 applications of streptomycin in a season. And those applications are put on at bloom when the trees are most susceptible to fire blight infection. And then pears, another crop that's highly susceptible to fire blight. We're seeing about 10,000 pounds of streptomycin use per year. Treated acreage is higher, can be as high as 60% or more of the acreage in California. Again, 1 to 3 applications of streptomycin. So the total annual usage of streptomycin on plants, this covers apple and other crops, and these data are from the 1990s. The range goes from about 20,000 to 65,000 kilograms per year, depending on the source. The lower estimates from the USDA, again, the higher estimate from the U.S. Geological Survey. But by either estimate, this usage is less than 0.5% of the total antibiotic use in the U.S., counting animal and human uses. So the use level is quite low. I think the one difference that we have, the one obvious difference, is that when streptomycin is applied to crops, for example, in an apple orchard, then the spray a streptomycin solution is sprayed from a sprayer onto the tree. So the antibiotic itself is broadcast into the environment from a sprayer. So that's the one difference in terms of the application. Okay, so let's talk a little bit about fire blight disease. Again, this is probably the most important disease in the U.S. on apple and pear trees. On a yearly basis, it can reduce fruit yield, but then the bigger issue is it will kill branches of the tree, and it can kill entire trees. So if we think about apple production, we've got rows of apple trees in an orchard. It takes about 3 to 5 years for an apple tree to come into production. And so every tree that gets killed by fire blight then represents a loss of production at that spot for a period of years for a grower. And so these are some symptoms from fire blight. Those are apples there in the left there with the bacterial infection. And the droplets coming out are ooze. That's the pathogen that's encased in a biofilm. And then that pathogen in those ooze droplets will spread between plants and within the plant from there. On the bottom is you can see shoots that are killed by the pathogen, and after the shoot is killed, then the pathogen can move internally through the tree down to the roots and can kill the roots, and that can kill the tree. So in the year 2000 in southwest Michigan, we had a major fire blight epidemic, and that resulted in about 450,000 trees killed, and of significance, that was about 1/5 of the apple acreage in the state was lost that year to fire blight. We had an overall 35% yield reduction, $42 million direct loss. So these are significant economic issues that growers face, for example, in apple production, and because of the high cost in terms of disease and also the high value of the crop, that has led to the ability of those growers to use antibiotics. Again, antibiotic use in plant agriculture is economically driven to a large extent, and so antibiotics are only used on the highest value crops and on those crops where bacterial diseases can cause issues such as what we see here that can really lead to significant yield problems. So fire blight is an excellent match, I think, between a plant disease system and use of an antibiotic for control. Again, it's a high-value crop, but the second point is that there's a focused time frame where the antibiotic should be used, and that's during bloom. So when the apple trees are blooming, that's when they're most susceptible to disease. That's a period of only usually about 2 to 3 weeks, and it's early in the season. So when antibiotics are used on apple flowers, pear flowers, for example, for disease control, then those residues do not show up in the fruit at the end of the season because the use is much earlier in the season. And the other aspect of that flower use is we need that significant population reduction for successful control, and the antibiotics are the only control agents that we've found that can provide that reduction. So this system is amenable, for example, to the use of streptomycin. But then the last point's important too, is that research over the years with fire blight disease, we know the temperature and the weather conditions that are required for disease to occur. And so we have two very good online weather predicting disease forecasting systems that growers can use. And these systems are in place for growers to know when and if they need to spray. So there are some years when conditions don't warrant any sprays, and so the growers can learn those from the forecasting systems, and then they'll only use the materials when they're most needed. So here's a photo of a flower. The stigma surface of the flower is the site where the bacteria grow, and they grow again to about even 1 million cells per stigma. So the stigma is shown by the top arrow there. That's basically a petri dish for this bacterial pathogen, and it lands on that stigma surface, and then within a day, if the weather conditions are conducive, then those populations can build from 100 cells to 1 million cells on one flower. And then if you think of the number of flowers on a tree in an orchard, we can have a tremendous population explosion. And so antibiotic use is targeting the organism at the stigma and then at the base of the flower where the pathogen will infect. So that's where we need coverage with our antibiotic solution to kill the organism and provide disease control. If we don't, then blossom blight infection is what occurs, and then the bacterium will move internally within the tree, and then it can't really be controlled very well after that. So, but if we do control blossom blight, then growers typically are in very good shape for disease control throughout the season. And this slide just shows some of the work that we've done at MSU, working with streptomycin for blossom blight control. And in these tests, what we do is we run field tests on susceptible apple cultivars, and we inoculate these trees with the pathogen. And so then we put streptomycin sprays on before and after we inoculate, and you can see that we can get infection as high as 80 to 90% infection in unsprayed trees. But then where we use streptomycin, that population or the disease is reduced very effectively and very consistently. And I guess that's the other aspect of streptomycin use on plants. The control is very consistent from year to year if we don't have resistance. And so this control with strep is used very widely in the U.S., in the West, Pacific Northwest, in the Midwest, and in the East. And then the 3 states that I've highlighted are the top 3 apple-producing states in the nation. Washington is by far the number 1 state. New York, number 2. Michigan, number 3. But these are the main states where streptomycin is used. And again, growers will typically put on 1 to 3 applications per season during bloom. Now, there are some issues with some of these highly susceptible apple cultivars that we grow. Even if they are protected from the blossom infection, they can still get shoot blight infection, which is what I'm showing here. And especially on young trees, this is really important because the young trees seem to be more susceptible to being killed after they get shoot blight infection. And so that can happen, and it is a problem. And in some regions of the country, what happened was the solution that growers had applied to this situation was they increased the number of applications of streptomycin that they used. And this is one of the categories that we would call a misuse of streptomycin, and I think you can tell what happened from that. And what did happen was we had streptomycin resistance issues in the West and Pacific Northwest, and then also in Michigan. And we've seen resistance in our largest apple growing areas in Michigan, the Southwest, which is shown by that black circle in the 90s, the Fruit Ridge area around Grand Rapids in early to mid 2000s, and then up to the northern Michigan in 2012. We've seen resistance. Now, in the Pacific Northwest, streptomycin resistance is conferred by a chromosomal point mutation. In Michigan, we found that resistance is gene-based, so we do have streptomycin phosphotransferase enzymes that are responsible for resistance development in the fire blight pathogen in Michigan. So because of that resistance, we went back to a second antibiotic in plant agriculture, Oxytetracycline. And this one, Oxytetracycline, structure identified in 1953. It has a wide range of uses across all medical, animal, and plant now. One of the issues with it is it's bacteriostatic, so it doesn't kill the cells. For fire blight, it can provide protection at blossom time, at bloom time, but it's not as effective on other diseases, so that limits the use of oxytetracycline on plants. Another aspect of use of oxytetracycline, for example, and use of any antibiotic in plant agriculture is it's affected by environmental conditions. So oxytetracycline is highly susceptible to being degraded by the UV component of sunlight. And shown on the bottom line in this graph, you can see within 2 days then the oxytetracycline dose is down to less than 10% or 20%, sorry, of its activity due to just environmental degradation. So use is impacted, and I believe it's not used as heavily on apple and pear because of that. It's less effective than streptomycin, and it's also impacted by environmental issues, environmental degradation. Oxytetracycline does have a use on peaches, though, against a disease called bacterial spot. And so if we look at its use pattern, it's very similar to streptomycin on apples, a range from about 12,000 to as high as 25,000 pounds per year. But there's more of a use on peaches than on apples and pears. On apples, for control of fire blight, oxytetracycline is less effective than streptomycin is. So this slide shows another test that we had done, and you can see that the control of blossom blight with Oxytet falls in the middle, and that's a problem because then, again, once we get that blossom infection, that can lead to increased shoot infection. And so oxytetracycline was not a great substitute for streptomycin for us in a situation where we had streptomycin resistance. Again, our optimal substitute would be plant host resistance in the tree, but since we don't have that, right now we've tested a myriad of other alternatives, but the antibiotics fall to be the best alternatives at the moment in terms of fire blight control. So we've turned to an antibiotic called Kasugamycin as a substitute for oxytetracycline. Kasugamycin is an aminoglycoside. It's in the same class as streptomycin. It was discovered in Japan, and it's very similar to streptomycin in its mode of action. It targets the bacterial ribosome. It does target a site that's different from strep, so there's no cross-resistance there if we had a chromosomal mutation. I think the important aspect of Kasugamycin use is there's no medical uses, there's no uses in animal agriculture to date. In fact, Kasugamycin has been tested against some human pathogens, for example, and it's not effective. It doesn't have efficacy against them. And in fact, it's not effective against a number of different bacteria, but in terms of Erwinia amylovora, the fire blight pathogen, it's highly effective against that organism. So it's an interesting use for us, I think, in that we think that there's not a heavy selection pressure put on in the environment when we use Kasugamycin because, it doesn't have efficacy against a lot of bacteria, but it does against the Erwinia pathogen. Kasugamycin We've tested it for several years now against fire blight, and in all these tests, this is similar to the slide I showed on strep. Kasugamycin shown in green, streptomycin in black. The control is not significantly different between strep and kasugamycin. So it works very effectively, very similarly to streptomycin. Now, one thing I forgot to mention at the beginning, which I should have mentioned, is that the use of antibiotics in plant agriculture is regulated by the EPA. And kasugamycin, or the compound Kasumin, has been used in Michigan. The EPA has granted what's called a Section 18 specific exemption. And so what that is it's an allowance for the use of kasugamycin in Michigan only in counties where we've detected streptomycin-resistant Erwinia amylovora. So it can only be used where we know we have resistance. And I think more importantly, it can only be used when the fire blight disease model predicts epidemic conditions. So there are years when the model would predict fire blight conditions but not epidemic conditions, and then Kasumin could not be used under those conditions. It can only be used when epidemic conditions are there. Can only be used during bloom. Can't be used more than 2 consecutive times and only 3 maximum applications. So Kasumin it's been adopted. The growers have been kind of slow to adopt it because it's very expensive. It costs twice as much as streptomycin does. So that's another limiting factor for apple growers in its use. They're only going to use it on their most highly prized varieties, the ones that will give them the highest economic return. And so, again, there are many other non-antibiotic methods of plant disease control that have been tested for bacteria. Copper sprays, biological controls, bacteriophage, for example, antimicrobial peptides, plant growth regulators, compounds that induce a resistance response in the host. Unfortunately, none of these are close to being as effective in the fire blight system, for example, as streptomycin or Kasumin. And so that's keeping their uses on the lower end. And it's keeping us on the end of using antibiotics. But again, the use is relatively low compared to other agricultural uses. We have 3 antibiotics that are used: streptomycin, oxytetracycline, kasugamycin. Again, the targets are high-value crops, and in these crop systems we do have stewardship-type mechanisms, disease forecasting systems that help to limit applications, and I think growers are becoming more and more aware of resistance management issues, especially with the onset of streptomycin resistance in the fire blight system.